Dear organization team, dear participant, first of all, I'm very happy to be here with all of you. My name is Narimane Segueni. I'm professor at the Faculty of Medicine, University Saleh Boumediene, Constantine III, Algeria. My talk today is entitled Algerian Propolis as an Effective Food Preservative, Antioxidant and Antibacterial Activities and Evaluation of Oral Toxicity. Natural preservatives have been the subject of interest of food industry. They are considered as a good alternative to synthetic one. They are more accepted by the consumer and are in most cases multifunctional material, offering the use of them instead of a combination of different organic compounds. Bee products are a valuable and rich source of biologically active substances. They have been used for centuries as a food and in traditional medicine. Among bee products, we choose to study the propolis. This amazing bee product has attracted our attention many years ago, and we choose to carry out several studies like this one. So propolis is a natural resinous complex material produced by honeybees from substances collected from birds, leaves, and exudates of different plants and trees. Propolis has a long history of use in traditional medicine, and several studies and several researchers all around the world have demonstrated that propolis possesses a large spectrum of biological activities such as antibacterial, antioxidant, anti-cancer, anti-inflammatory, anti-biofilm, immunomodulatory activity, and many other activities. Regarding chemical composition, propolis chemical composition is quite variable, dependent on the provenance of the sample, which is strongly related with the flora surrounding the hive. Therefore, variation in propolis bioactivity in general are expected to be associated to the chemical diversity of the constituent. We reported in the present study our investigation on the potential use of Algerian propolis as a food preservative. The propolis studied was collected from beekeepers located in the northeast of Algeria in the wilaya of Jejel, as you can see here in the slide. By scraping frames, walls, and the entrance of the beehive and stored away from light and heat until it was ready to use. First, ethanolic extract of propolis was obtained according to the method described by Park. Then the antibacterial activity was evaluated using agar diffusion method and the determination of minimal inhibitory concentration. Antioxidant activity was also evaluated using 4 different assays. And finally, we evaluate the toxicity of propolis on 2 functions, renal and hepatic function, after an oral administration of a daily dose of 300 milligrams per kilogram of body weight of propolis extract in watt during a treatment with 2 weeks. Phenolic acid composition was also evaluated and determined using ultra-fast liquid chromatography coupled to tandem mass spectroscopy. And you can see here in the slide the different parameters we used, such as the column, the gradient of mobile phase, the injection volume, the flow rate, and all other parameters. All experiments were performed in triplicates, and one-way analysis of variance was used to determine the difference between the tested parameters using the adequate software. As you can see in figure 1, the studied propolis was found to be very rich on phenolic and flavonoid compounds. And these results is with agreement with our previous investigation on propolis collected from the northeast of Algeria, which was all found to be very rich on phenolic compounds. They are probably poplar type propolis. Regarding the phenolic acid constituents, our results indicate that the main constituents of this propolis are caffeic acid, chlorogenic acid, gallic acid, ferulic acid, and also rutin. Other phenolic acids were also identified in the extract, such as pegfumaric acid, vanillic acid, and syringic acid. 3 gram-negative bacteria associated to 1 positive bacteria have been used for the evaluation of antibacterial activity of the tested propolis, and the minimum inhibitory concentration was performed using the agar-dilution method following the clinical and laboratory standard institute guidelines. And as you can see here in this figure, the studied propolis was found to possess more pronounced activity on Staphylococcus aureus compared to the other bacteria. And this propolis possesses a weak activity on all gram-negative bacteria, in particular on Klebsiella pneumonia. A strong and positive correlation was detected between the total phenolic and total flavonoid content and the MCC values of Staphylococcus aureus, while a weaker positive relationship between Klebsiella pneumonia and total phenolic and total flavonoid was observed. Regarding Escherichia coli and Pseudomonas aeruginosa, a medium negative correlation linking minimum inhibitory concentration and total phenolic was observed, and a weaker negative correlation was observed for total flavonoid content. The results reported in the present study are in accordance with our investigation on Algerian propolis. We previously investigated a lot of propolis collected from the same localities in Algeria, in the northeast of Algeria, and all the studied propolis were found to possess a more pronounced activity on Staphylococcus aureus compared to other gram-positive bacteria that we studied before. The study are not reported here in the present presentation, and all the studied propolis also were found to possess a weak activity on gram-negative bacteria. It seems that Algerian propolis, especially those located in this region, act as an anti-staphylococcal drug, and further research are in need to understand and link this activity to the chemical composition responsible for this activity, and also to try to explain the mechanism involved in such activity. Recent studies confirm our finding regarding the antibacterial activity of propolis, which was reported to be more pronounced against gram-positive bacteria compared to gram-negative bacteria. This activity was attributed to different in-cell wall structure and the presence of synergetic active compounds such as flavonoid and phenolic acids were responsible for this activity. Some compounds were appointed to be responsible of the activity, such as atripylene, C, quercetin, and pinocombrin. The three compounds possess a significant inhibitory effects on pathogens, including Staphylococcus aureus and Enterococcus faecalis, with some flavonoid enhancing the activity of conventional antibiotics through synergetic interaction. We performed other study on this, the same propolis, which we are talking about today, and in the chemical investigation of this propolis, using the different spectroscopic and the different chromatographic analysis, we were able to isolate and identify other compounds than those reported here. And those compounds were identified as responsible for other properties. But among those compounds, quercetin and pinocombrine were isolated in this propolis, and in all other studies about the propolis located on the northeast of Algeria, we found the same constituents, and quercetin and pinocombrine are main compounds of those propolis. As you can see in this figure, the studied propolis was very important and interesting because it also possesses an interesting antioxidant activity. It acts highly on the cuprac assay and possesses a weaker activity when we use FRAP assay. The present findings suggest that propolis possesses the ability to scavenge radical by acting as an electron donor. In addition, propolis possesses reducing properties, probably due to the presence of reductants in the tested extract. And finally, we study the potential toxicity of propolis after an oral administration. Our study was conducted on female Western rats weighing approximately 170 grams. Before and after treatment, the animals were raised in plastic cages, divided into 3 groups with free access to food and water. Our study was conducted over 2 weeks. Each animal received a daily dose of the appropriate treatment. The animals were divided in 3 different groups. The first group received physiological water. The second group received ethanol solution. The third group received our treatment with a daily dose of 300 milligrams per kilogram of body weight of fat during a period of 14 days. Then the blood symbol were corrected at regular interval, as you can see here in the slide. The blood was treated to obtain the serum, and the serum was stored in a freezer until biochemical analysis were performed. The animals were dissected at the end of the treatment, and the liver and the kidney of each animal were removed and weighted. We evaluated the liver function using the different assays you can see here on the slide. We also evaluate the kidney function using the different tests you can see here. And our results indicate that the treatment of animals with ethanolic extract of propolis at a daily dose of 300 milligrams per kilogram for 2 weeks is devoid of any toxic effect on renal and hepatic function. However, for further study with other biochemical parameters, as well as a histological or histopathological study of the 2 studied organs are necessary to confirm these preliminary results or these first results reported here in the present study. So, as a conclusion, the studied propolis was found to be more effective on Staphylococcus aureus, and this propolis possesses also a very interesting activity. In addition, it is devoid of any toxic effects on the renal and the hepatic function. This finding is very important because the effect on Staphylococcus aureus suggests that the use of this propolis can improve food safety and extend shelf life by inhibiting this common and harmful foodborne pathogen. Staphylococcus aureus is known for causing food poisoning through toxin production, and its presence in food products poses significant health risk. The reported antibacterial and antioxidant activity can help delay lipid oxidation and preserve food quality. Propolis can be considered as a good food preservative and can be used in the food industry, especially for managing staphylococcal contamination. As a natural preservative, propolis can be applied in various food products, including meat and dairy, to reduce microbial contamination, supporting safer and longer-lasting foods. Before to leave you, I want to show you some picture of my city, Constantine. Constantine is one of the biggest cities located in the northeast of Algeria. And Constantine is known for its bridge, as you can see here in the slide. For any contact, you can send me email using one of my emails you can see here. And finally, thank you for your attention.